Hello and welcome to our third webinar this year, brought to you by Genomics. Our mission at Genomics is to pioneer advancements in 3D tissue culture, presenting an ethical, economical, and highly predictive alternative to traditional cell and animal testing. Today, we have the absolute privilege of hosting a leading figure in the field of biomaterials, tissue engineering, and regenerative medicine, Dr. Miguel Castillo. He serves as an assistant professor of biomaterial design and processing at Eindhoven University of Technology. Holding an MSc in mechanical engineering and a PhD in biomedical engineering from the University of Lisbon, his expertise lies in bioactive, inorganic material design and 3D printing. Miguel's research focuses on the development of design and fabrication technologies for the local control of biomaterial composition and structure, specifically for musculoskeletal tissue regeneration. He combines advanced 3D printing technologies with computational design to develop instructive materials for regenerative medicine. A very committed educator, Castillo is also a lecturer on biomaterials, tissue engineering, and regenerative medicine technologies. He's also a very prolific author, had made significant contributions to biomedical engineering, really high impact papers, and lots of them as well, particularly in the realm of biomaterials and 3D bioprinting. He's also an active member of several professional bodies, including TERMUS, the ESB, and ISBF. And we've got a pleasure of hosting Miguel's talk today, which is entitled Electrowriting of Cell-Laden Natural Derived Polymer Fibers. So in this talk, he will be talking about the development of functional fiber replacements that can promote in vivo repair of musculoskeletal tissues. He'll also explore recent advances in electrowriting 3D printing of natural polymers and showcase examples of electrowritten microfiber scaffolds and discuss the functional properties of these silk-based scaffolds as well. So before we get started with our guest presentation, just a quick few housekeeping items. If there's any technical issues at all during the presentation or even afterwards or before, please let us know via the chat function and our team will try to rectify those as soon as possible. Also, we encourage you to, of course, engage and ask as many questions as possible during and after the talk as well. To do so, please use the questions section on the GoToWebinar interface. You should see that on your right-hand side of the screen. So yeah, feel free to type in a couple of questions during the talk already. And once Miguel is finished with his presentation, we'll have a couple of minutes for Q&A as well and discuss his findings and his presentation today as well. So without further ado, again, thank you so much, Miguel, for joining us today. It's really a pleasure to have you here today and host the presentation. And yeah, we'll probably just get started to share your screen. And yeah, we're looking forward to your presentation today.
Oh, fantastic. Thank you very much for the kind introduction and for having me.
Just 1 second, just trying to get, here we go. So
can you see it now?
We can see it and we can hear you as well. Thanks, Miguel.
Okay, fantastic. Let me just move this down. Okay, once again, thank you very much for having me. It's of course a pleasure to be here and then with you and then present some of the latest work that we have developed throughout the years in my research group. So as many of you know, with the aging and the changing of lifestyles that are driving a rapid increase in the chronic disease burden around the world, our healthcare systems are basically being geared towards caring the chronically diseased to relieve symptoms and then slowing down the progression of the disease. And this is basically exactly why regenerative medicine and materials that can promote diseased or damaged tissues regeneration instead of simply repair can make a powerful contribution. And this is particularly interested to address health challenges like cardiovascular diseases, kidney failure, arthritis, and osteoarthritis that on their own cannot heal themselves. So in my team, we are particularly interested on generating fibrous tissues. And the reason for that is that the majority of you know that our living tissues are composed by cells surrounded by an extracellular matrix. And one of the major components of the ECM are basically fibrous proteins, such as collagens, elastins, and fibronectins. And these fibrous proteins are basically synthesized and secreted by the cells, and then assembled into large fiber structures at or in the extracellular space in the presence of other macromolecules. And these fibers, components of ECM, are basically responsible to provide structural support and integrity to different tissues and organs also in our body. One aspect is that depending on the tissue type, of course, these fiber structures not always regenerate. And this is particularly crucial, for example, to collagens. And I give you an example of collagen in articular cartilage that, when disrupted, it can actually not regenerate on its own. And this is also applicable to other tissues like tendons and ligaments, and also in the cardiovascular fields to the myocardium tissue. So this is exactly why in our group we are basically interested on the investigation, design, and fabrication of fiber structured materials that can guide tissue regeneration. And then we do this by focusing on three aspects. The first one is to develop and advance fiber-based processing technologies that would allow us to create fiber scaffolds. And then we explore these fiber scaffolds, or we use these technologies to explore at two different levels. One level is to basically develop fiber materials that can imitate complex tissue and organ functions, and I will provide you a couple of examples later during this presentation. And another area of research is to understand how actually these native fiber proteins are naturally formed, how they are secreted by the cells, what are the mechanical forces that drive these, and in which conditions they move from simple fibrils or molecules that assemble in large fiber bundles. So this understanding is something that we are very interested in. So how do we go from here, and how do we approach this challenge? So we basically try to combine the bottom-up with the top-down approach. At the bottom-up approach, we study the physicochemical environment at which these fibrous proteins assemble. So starting from the molecules up to fibers, and then the top-down approach, we basically explore how we can upscale it to large fiber bundles at the macro scale and control organized scaffolds that have differential organizations, control porosity, and pore geometries. So really the top-down gives us the control over what we call it the micro and the macro structure of these fiber scaffolds, while the bottom-up provides us control over the assembly from the macromolecules to small fibrils and fibers. And we try to combine these two. We predominantly look at nozzle-based printing techniques like extrusion-based and electrowriting, because those, we believe, provide the mechanical environment to confine and organize fiber proteins. And this, we believe, it's very similar to what it happens in a living cell, because, for example, collagen, it's secreted by creating invaginations in the cell membrane that basically create a kind of a shear force in there when laying down the collagen. And then these collagen fibrils, in the presence of other macromolecules in the extracellular space, they basically then self-assemble and creates large fiber bundles. So one of the areas that we are particularly interested, as I mentioned, is to understand in which conditions these fibers or fibrils, to be more precise, assemble. And it's known that from the biological processes that self-assembly of fibrous proteins take place in extremely crowded environments. So typically our ECM environment, 5 to 40% of volume is occupied by other macromolecules that participate in this self-assembly. process. And to investigate this, basically, we took synthetic polymer, in this case polyethylene glycol, and then we combined it at different concentrations with collagen molecules. And what you can see here from the bottom is that we wanted to be the measurement. So basically, we look at the speed at which these collagen fibers will self-assemble in the presence of different concentrations of PEG. And what you observed is that at high concentrations of PEG, as expected, the process of self-assembly, we call it fibrinogenesis, was way faster in the range of a few minutes, while compared with simply collagen in an environment with specific pH and temperature without the crowding agent. And then we confirm this with scanning electron microscopy images or analysis, showing the images here on the left side, where you basically see that at the higher concentrations of the PEG, the crowding agent, it led to a highly dense network of collagen with basically a high density of thinner fibers of collagen. So typically the crowding agent plays a huge role, and we can somehow mimic or approximate to the conditions that this happens in living tissues. The next step was basically from the top-down approach, combine this principle of macromolecular crowding with a process. In this case, we picked extrusion-based printing. And the reason for that is that we believe that the nozzle, and by shearing the material throughout the nozzle, we can further confine and promote the assembly and entanglement of the protein molecules. So we didn't want to explore any chemical modifications to our collagen, or in this case, because we believe that that will have harmful impact later on. So we basically just took collagen in acidic conditions. We combined it with or not with the crowding agent, and then we extruded this in a support bath composed of gelatin at neutral pH. And then this shift between an acidic ink in contact with the gelatin bath at pH, the neutral pH, basically led to inducing the fibrinogenesis and self-assembly of collagen. And then this gelatin, as most of you know, can be removed by just increasing the temperature, so basically melting the gelatin. So how the process goes, you can see here in this movie, and it's already an established process in the field. The main novelty now is the inclusion of the crowding agents in our ink. You see basically the nozzle traveling inside of the support bath composed of the gelatin granules that basically provides support to the liquid-like ink that we are extruding. And then by repeating layer by layer, we could achieve our 3D scaffolds. It was very interesting for us to see that when we compared structures printed with or without the macromolecular powder, we observed that in the presence of the macromolecular powder, we achieved basically denser and more aligned constructs. So you can see here by these scanning microscopy images of one single filament or one section of the printed construct. So typically, this was the contribution of the macromolecular crowding. And what we also observed is that independent of the presence of the crowder, we could print collagen that showed the main hallmarks of collagen. So basically the debanding structure and the fibril network, which is typically not trivial to achieve when we started from simple diluted collagen molecules. So we can now process collagen and achieve the main characteristics of collagen, being the debanding pattern and the fibrillarly network, and with the presence of macromolecules, or in this case a synthetic polymer PEG, we could further densify and achieve some sort of a more aligned printed threads. What we are doing now is to further investigate if these constructs are cross-linked or not, because the reason for which collagen in our body does not dissolve is because they are cross-linked, right? Otherwise, they will just dissolve. So we are investigating in which form is the collagen that we are printing. And then we are using these collagen scaffolds to template processes like mineralization in order to generate and produce a piece of synthetic bone. And we believe that achieving this collagen density that presents the main hallmarks of the native collagen, it's crucial not just to the regenerative process, but also to the mechanical function of the collagen, and also to template processes like mineralization. Another area that that my team is interested, it's on basically investigating strategies to regenerate coordinated contraction in cardiac muscle after cardiac damage. So as probably some of you know, our heart, it's composed of collagen fibers organized in an isotropic way, but in a kind of a very unique organization. So basically at different length scales, and the collagen fibers are typically organized in a basket weave, as you see here from the bottom images, that allow the heart to have a motion of torsion and pumping that will allow our blood to be pumped out of the heart and get into the heart. And it's basically this organization of the collagen fibers that you can see here from the MRI image at the bottom, that it's organized, as I mentioned, in this basket weave organization, so highly anisotropic, that allows the myocardium to function and provide this pumping and torsion function that is crucial for the functioning of the heart. The problem is, in the event of an infraction, this organization is lost. So the heart has a capacity to immediately heal that piece of diseased or infracted heart, but it creates a type of tissue that is known to be fibrotic. So it's basically composed of fibers, but highly disorganized. So losing the organization basically leads to a loss of contraction or contractile behavior of the heart and function, respectively. So what we tried to do was basically to explore the use of another nozzle-based printing technique, in this case, what we call the electrowriting. So basically a widely known technique probably for some of you, and that basically merges the principles of extrusion-based printing with electrospinning, but in this case, we are not dissolving any polymer. So we are basically just melting a polymer. So combining the forces of pneumatic extrusion with an electrical field, we can achieve very thin polymer filaments that have diameters that span from a few microns, from submicron scale to a few microns, like 10, 20 microns. So in this case, we look at hexagonal shaped scaffolds that we combined with IPS-derived cardiomyocytes, and then we investigated the mechanical function of these constructs and the effect of geometry on restoring seeded cardiomyocytes maturation and organization. The reason for looking at these hexagons is that in this case we were looking at polycaprolactam, which is an FDA-approved polymer that has all the benefits in terms of later clinical translation, but it suffers from some mechanical pitfalls. So at large deformations, typically above 5%, this material, the bulk material, basically undergoes permanent deformation, which means that if you cyclically stretch it, it will basically permanently deform, which is, as you can envision, it's something that you don't desire as a material to replace a piece of your myocardium tissue. So what we did was to keep this bulk material property, but play with the design. And by playing with the design, we wanted to achieve the stretchability or the elastic deformation that would allow us to have a patch that could deform elastically within the range that it's needed for myocardium repair, so typically between 20 to 30, and still show elastic behavior without, of course, undergoing permanent deformations. And the hexagons actually allowed us to do that. And what you see here from the bottom right side of the PowerPoint It's basically a comparison of printed hexagons with different pore sizes under unique cell tensile loading with the same PCL printed, but now instead of printed in an hexagonal geometry, printed in a rectangular geometry. And what you can appreciate is that the rectangular shaped constructs above 5%, and I don't know if you can see my pointer or arrow, it starts to undergo plastic deformation. While the hexagons, basically they went up to 30 to 40% within the elastic regime. So clearly, by just playing with the design, we could adjust the elastic properties of our constructs. We then went further on investigating how the structure could eventually restore organization or not, or induce maturation and induce alignment of cells, and later on to restore organization of the damaged tissue when implanted. We performed immunofluorescent stainings at day 14 of culturing of IPS derived cardiomyocytes in these printed PCL scaffolds. And what we showed is that in these confocal images, as you see here, is that the hexagons showed enhanced alignment, as seen basically by the sarcomere content, as stained by the alpha actinin in green. And we also looked at connectin 43 protein, which is expression, which is basically a protein that is present throughout the cardiac patches in this case, and then basically tells you if they support electrophysiological coupling or not of the IPS derived cardiomyocytes. And this is stained in red. And then we compared the cells combined in the two geometries. We also went further on quantifying the sarcomere length. And I think it's clear from the confocal images that in the rectangular constructs, they showed they were more disorganized, while in hexagons, they were more organized towards one preferential direction of the hexagons. And I should actually mention here that we were not printing perfect hexagons. We are printing more deformed hexagons that show the chain link type of organization. And that, of course, induced the alignment. So the sarcomere length was longer in the hexagons. They were more aligned. And as a functional parameter, we quantify the contraction rate, and then we observe that in blue, it's the contraction rate of the hexagonal shaped patches, that the contraction rate was higher. So the synchronous contraction of the cells, so they were able to couple and then contract in a synchronous way, was higher in the hexagonal constructs. We went one step further and then investigated, given these elastic properties of the constructs, could we then explore the use of these constructs towards a minimally invasive delivery, since they show this elastic behavior? So basically what we did was to basically roll these constructs and then deliver them through a small catheter. And these are basically the images that you see here, delivered in a beating peak heart. And as you can observe, basically the entire scaffold, when in contact with the heart, the wet layer of the heart was unfolded, fully unfolded, and then it was in place for the time that we did this test, which was basically just 3 days, and cells were alive. So basically the scaffold showed the shape recovery properties. It was able to unfold and then somehow align. And this project was basically at the base of an European grant called derived. We are now further exploring these designs and then studying how can these constructs integrate with the native heart, how can they synchronize with the beating of the native heart, will it interfere or not, how can we ensure that they adhere and then stay in place over time. So all these aspects are being now further explored and investigating, and this was just a preliminary test showing us some promising behavior from these patches. One of the challenges that we still face is the fact that we are trying to basically restore the contractility function of the heart. And this basically relies on the units, so the cells that provide the contraction. So they are basically the engines that actually contract. What we observed is that for the thickness of the constructs that we have, the contractile force that we generate is not sufficient to replace the contractile force that was lost in the infracted region. So what we thought was, one aspect was, of course, scale up the constructs, so make them thicker. But when you make them thicker, one of the challenges, of course, is vascularization, because the cells consume highly nutrients and oxygen. So vascularization starts to kick in as a very challenging problem that we have to address. On the other angle, we try to keep the thickness of this construct with the same cell density, but now instead of printing PCL, print a material that can be extracutaneously activated. And in this case, we looked at magnetized polycaprolactone. And the thinking here was, if we can further boost the contractile properties of this patch, we can eventually get to replace the contractile force that was lost after infraction. So to do that, we look again at the hexagonal microstructures, and then we explored the reduced graphene, and graphene for the reason that it promoted myogenic differentiation, or it's described in the literature to do so, and has a low reduction in cell viability. But as we know, reduced graphene oxide, it's not magnetized. So basically what we explored first was to investigate how iron could be oxidized in the graphene nanopellets. And basically it was very interesting to see that through the chemical process that we observed, we could in situ deposit iron oxide nanoparticles on oxidized graphene. And they were homogeneously distributed in these particles. And what we did then was to compound these particles with PCL using a melt blending approach that allowed it to fabricate extruded filaments. And what you can see here from these images, so the second column at the bottom is the extruded blended PCL filaments, so polycaprolactone, with different concentrations of the iron oxide nanoparticles oxidized on the top of reduced graphene oxide, and they were somehow homogeneously distributed inside of the PCL, which is typically a challenge when you could create compounded materials with particles, is to get an homogeneous distribution of the particles. So by looking at melt blending, we could actually achieve so, and then load PCL with, or compound PCL with particles percentage up to 20%, which was quite good for us. The next step was then investigate how this compounded PCL material interfered on the melt writing process. And as you can appreciate here, we tried again to print the hexagonal shapes. It decreased the printing accuracy of the constructs, but still allowed us to achieve a kind of a chain link and distorts hexagon with reasonably good stacking of the fibers on top of each other, and with a good distribution of the particles throughout the printed polymer filaments. Of course, the next question was to understand what were the magnetic properties of these materials. So we compared constructs loaded with different percentages of particles from 2% up to 20%. And it was when we look at the magnetic hysteresis curves on the right side of the plot, we showed that when we compare the melt extruded or the melt blended filaments and the melt written filaments, there was no decrease on the magnetic moments. And all the constructs typically show, for those familiarized with the fields, a typical ferromagnetic behavior as is characteristics by these hysteresis curves. So the melt writing process was not basically detrimentally affecting the magnetic properties of our materials. The next step was basically looking at how the construct influenced on cell organization. And here we took a cell line that was more easy to work with. In this case, we look at C2C12 myoblasts that we can culture on these printed constructs. And what you can basically observe here is that the cells basically tended to align along the printed fibers, while that was not observed when the cells were cultured on a 2D platform or even in a different shaped 3D electrowritten construct. We also look at a myogenic differentiation marker, in this case the MWH4, and we observe that this was, and here it's basically stained in green, and in dark it's basically the nucleus of the cells. We basically show that this myogenic differentiation marker was significantly higher for cells grown on the 3D printed electrowritten scaffolds, while compared with the 2D cultures, as I mentioned. And that scaffolds actually favor the alignment along a single direction, as observed also in the printed, not compounded IPS cell seated in the scaffolds. One of the steps, of course, was to look at the magnetic responsive properties of these materials and then repeat the same assignment, so culturing cells in the scaffolds and then magnetically deform the constructs and then show if cells survive, stay in place, and if they show the same alignment. This is part of the work that we are still running, and as you see here from the top image are basically constructs with the C2C12 cells seated inside and then exposed to a magnetic field. So basically we use an electromagnet to either put on or off the magnetic field, and that basically induced the deformation of the construct. And then we look at the cell distribution inside of the construct before and after the stimulation. And what you see here from the bottom is that the cells were within the construct after, in this case, 3 cycles, and we went up to 10 of deformation, which was already a good indication. And what we are now investigating is, do they still align in the same way or not, and how do they react to this different type of stimulation and the presence of magnetic fields. So the results were promising, but so far what I've shown you was basically the use of thermoplastic materials for electrowriting, which basically suffer from permanent deformation, as I mentioned. The way to go around that was, of course, play with the design in order to get that elastic behavior, but that it's not always desirable because the printing process also has limitations in terms of the accuracy of the geometry that you can print with. So we have been looking at a different class of materials that could allow us more long-lasting fiber network fabrication and to perform in an elastic way under large deformations. So one of the candidates that we came across was silk for the known mechanical properties that are widely described in the literature. So silk is extremely strong and elastic, and then of course would provide an alternative to thermoplastic materials, in this case PCL. But one of the limitations when we process silk, or in this case when we extract silk, is that we destroy the structure of silk. In this case, basically the beta sheet structure and the molecular alignment of the silk molecules, which basically then imparts the elastic performance of silk. So one of the challenges is to come up with a process that when we extract it with the existing processes, like taking in this case the silkworms from a cocoon, digest the cocoons, isolate the extract the silk fibroin, and then we get rid of the sericin, was to how do we restore the beta sheets and organization of the silk molecules in order to achieve the same elastic behavior and strength that is characteristic of silk. We advanced the principle of electrowriting towards an aqueous electrowriting process. So here we created basically a nitrogel type of silk fibroin ink, and then we investigated how it can be processed under room temperature using in the presence of electrical fields in order to achieve the same small fiber diameter polymer filaments, and keeping the same ability to pattern, so achieve an accurate design of your printed constructs, because that is quite relevant, as I introduced to you, for all the aspects of restoring organization, and further dive in on the molecular organization, if I can say it like this, of the silk, and check how the process, how can we restore basically the beta sheet formation and the molecular alignment at the nano scale. So as you can appreciate, AquaSilk solutions are quite tedious to work with in terms of the rheological insulation behavior. So it's quite difficult to control them. So basically what we did was to explore the different boiling time of extraction. So we start from silk cocoons, and then we digest these cocoons in order to extract the silk fibrin, and control the molecular weight, and with the molecular weight control the viscosity, because viscosity is a crucial parameter for electrowriting. So there are basically three aspects that we should consider when developing materials for electrowriting. It's the rheological properties, mostly the viscosity, the electrical conductivity, because we are applying an electrical field, and the thermal stability of these materials. In order to control the viscosity, since we were looking at an aqueous-based material, we played with the boiling time, so the molecular weight and respective viscosity, and we also added viscosity enhancer, in this case PEO, that has a low electrical conductivity. And following a design of experiments, we concluded that there was an optimum in terms of the boiling time and the PEO concentration that would allow us to create a stable jet formation that was not basically dripping, and would allow us to achieve a continuous collection of the filaments. And basically this led to achieve structures, and what you see here are basically some images of the printed structures. In this case, we look at flat and tubular, and we could lay down silk fibers up to 100 layers. And typically the fibers had around 10 microns of diameter, and we could lay down with quite a good accuracy up to 100 layers in a controlled way. Of course, there is always some misplaced fibers that it was hard to control, but this is also due to the nature of the natural polymers that are quite tedious to work with, and it's not always easy to control their properties, even if you control all the extraction process and all the processing conditions. So there is always some heterogeneities in your ink that led in the environmental conditions that led to these misplaced fibers. But overall, we were quite pleased to see that we could achieve flat constructs with a square-shaped architecture up to 100 layers, and we could also achieve a tubular-shaped construct with the same layers, in this case tubes with a 4 millimeter inner diameter, and again 100 layers stacked down on top of each other. And here we look at winding angle, basically the angle between the printed walls of 90 degrees. So this was basically processing the silk with electrowriting, but one of the challenges was, of course, these materials did not show significant restoring of the beta sheet structure. So basically what we did then was to investigate a post-processing treatment. In this case, we look at sodium phosphate, because it's known that in the presence of some ions, if you think on silk from spiders, when they lay down the silk in the spider ducts, silk molecules are exposed to different concentrations of ions, and that it's known to induce the cross-linking of silk. In this case, we selected sodium phosphate, and we compared the printed constructs treated with sodium phosphate versus the conventional method, in this case methanol. And what we observed is that with the sodium phosphates, it was able to induce physical cross-linking and apparently the formation of beta sheet structures, as observed here by the Raman analysis and the DAR. And also went further on trying to understand if from the molecular organization it was any evidences that by shearing the material out of a small nozzle, we are basically achieving basically the organization of the, or basically the organization of the crystallites and the beta sheet structures inside of each printed polymer filament. We look at AFM, so atomic force microscopy, and we observed that there was kind of a shrinking and more striations on the sodium phosphate treated printed filaments that led to a more smaller diameter and more confined filaments, and that we attributed these to a more aligned construct or compared with the silk-only fibers not treated either with methanol or the sodium phosphate. Of course, this required still in-depth analysis. We are now looking at the cross-sections of the printed filaments to confirm if there is any indeed crystallite alignment, and we are also looking at other techniques like SACs to further investigate at the molecular level what is actually happening. What was without this in-depth understanding, what we could look at the micro scale is that the printed constructs treated with solid oxide showed an elastic behavior. So when we applied uniaxial tensile loading to these constructs at increased displacements, basically we showed here in this movie that basically the printed constructs being stretched that increased the strength, and we went up to 20%. For 30 cycles, there was no evidence of significant elastic deformation. There was a decay in the resisting tensile force, but not significant. So this basically told us that indeed the constructs, the combination of electrowriting with the treatment, the post-treatment with sodium hydroxide, seemed to induce a more elastic-like type of behavior that was confirmed by these mechanical tests. And this is possibly attributed to the beta sheet crystalline structure and organization of the printed constructs. We then also went further to basically, as I showed to the for the hard patches, that a possible delivery approach is to use to deliver this patch through minimal invasive surgery, so deliver them through a small catheter. We also explore the potential of these silk-based constructs for the same minimal invasive delivery strategy. And what you see here from the images at the bottom is that it can be folded and then fully unfolded when in contact with an aqueous medium. Two last applications that I want to show to you that we developed in our team. One is for kidney. So kidney, as you know, basically comprises 1 million of functional units that are called the nephrons. And then inside of these nephrons, they are basically composed of the proximal tubule. And this entity basically contributes to the hemostasis and then resorption of 60 to 70% of water, salts, and most nutrients. And then it's basically responsible for filtrating the urine. So one of the strategies, if you think on people that suffers from kidney failure, they basically have to undergo dialysis. And the dialysis membranes are basically a piece of plastic that filtrates basically the toxins that we want to get rid of, but also gets rid of a lot of components that we want to remain in our body. And the reason for this is that we are using a piece of synthetic plastic that basically does not function as in the same extent or far from the living proximal tubule. Why? Because here are the kidney cells, the kidney proximal tubule cells that do this filtration. So in the dialysis membranes, using a piece of plastic shows some promising results, but we also get rid of important components that we want to keep in the body. So we need typically to look at alternative strategies that would allow the kidney proximal tubule cells to be themselves to do the filtration. So Of course, we want to look at materials that are highly porous. So typically techniques like electrospinning will not be desirable here because we want a material to be first in the tubular shape, because that tubular shape is important for the cell differentiation and function of the kidney proximal tubule cells, and we want them to be highly porous. So that's why we have been looking at techniques like electrowriting that can create different highly porous structures with different control pore angles. In collaborations with colleagues from Utrecht, we look at Utrecht University, we look at printed tubular structures with different winding angles that span from 30 to 70 degrees. And then we cultured both conditionally immortalized proximal tubule epithelial cells on these and conditionally immortalized glomerular endothelial cells on these. And then we tried to find the magic angle that would allow to create a monolayer of cells inside of these tubes. And their organization, that it's also crucial for the function of these proximal tubules. So we succeeded on creating these tubule constructs with different winding angles, now out of polycaprolactone again material, not the silk as I introduced to you. What was interesting to see was that both the SIPTEC and the endothelial cells formed tight and homogeneous monolayers within the tubular scaffolds and generated collagen 4 enriched ECM matrix. I don't want to bother you with all the details, but what you can see here is that the 30 degrees widening angles induce the preferential alignment for the SIPTEC cells. And the difference between the three different angles, when you look at the confocal images on the right, was less pronounced when we cultured the endothelial cells. So it seems that there was no preferential alignment between the three different angles, although the 30 and the 50 degrees showed some sort of preferential alignment. And of course, these are preliminary results that we are investigating. And we also tried to combine an endothelial region with kidney proximal region at the center. So to do that, we looked typically at the seeding the endothelial cells at the outer part and the kidney cells at the inner part. And basically what you see here was basically we were able to create a monolayer of both cell types using the same scaffolds as a template. And we look at endothelial marker like the CD31 that was highly expressed in the seeded constructs, and you see here by staying in green. And we also look at the functional marker, in this case the Z1, which basically tells you about the tight junctions between the cells, and it's an indication of the filtration capacity of the cells. So it's possible to, so structure guides organization again. It's possible to create monolayers to seed kidney cells in these tubular constructs with organized architectures, induce their organization, and we can create basically a double region tubular constructs that can filtrate that shows promising to filtrate because we didn't show that. And then these, the compounds that we want to recover to the bloodstream can then be recovered by this endothelial layer surrounding the kidney layer. So again, promising results that we are further exploring. Another application was, up until now, we know that we culture cells in the presence of growth factors in the cell culture environment. And typically we use high physiological concentrations or physiological concentrations or concentrations of these proteins that are way higher than the physiological concentrations in the native body. So what we have been looking was at strategies to further functionalize these electrowritten scaffolds in order to guide tissue regeneration, but now using just a small amount of these growth factors. We look at the cartilage just to give you an example. And in cartilage, when the collagen structure is disrupted, it's highly being restored. In order to culture cartilage in vivo, we typically use TGF-beta because it's growth factor that it's known to promote chondrocytes to differentiate and to lay down, chondrocytes being the cartilage cells, to differentiate and lay down their matrix, in this case the collagen type 2 and the GAGs. So what we did was to take our PCL polycaplacton mesh, and then we teamed up with colleagues in Australia from the group of physics at the Sydney University, and then we investigated the technique called atmospheric plasma jets to activate the PCL fibers, and then explore if we could covalently attach or mobilize TGF-beta. It was very interesting to see. We explored different conditions, and then I will walk a bit quicker or faster here. We tested different conditions of functionalization. So basically, we had the plasma pen passing over the scaffold at different speeds, so a fast, a medium, and a slow. And then we look at the oxygen groups, oxygen-containing groups that were present at these different speeds, and we related this to the wettability of the material. And what you can see here from SPS analysis and the water content angle is that at the low speeds, medium to low, we could get more oxygen-containing groups present in the activated PCL meshes, and that led to more hydrophilic surface. We then investigated the attachment of TGF-beta, and basically what we did was to soak these meshes in the presence of a TGF-beta containing media, and then we stained, we washed the mesh with detergent. So if the growth factor was not covalently attached to the mesh, it was basically being washed out. And then we stained after this detergent wash if the TGF-beta was present there or not. And by our surprise, we saw that, as you see here, stained in green, using immunofluorescent staining for TGF-beta, that the plasma-activated meshes were able to covalently attach with the TGF-beta. And that this TGF-beta was, when we cultured chondrocytes on them, was able to guide chondrogenesis and matrix deposition in the same extent as meshes with chondrocytes seeded and TGF-beta supplied in the media. So basically what you see here on the right side are histology sections of the in vitro culture construct. So the meshes either with the TGF-beta attached, treated with plasma and TGF-beta covalently attached, or meshes with seeded chondrocytes and TGF-beta supplied by the media. And as you can appreciate here, so we look at two markers, collagen type 2 and GAGs, there was no significant difference between the groups. So this was an evidence that the TGF-beta covalently attached to the surface of the PCL mesh was active and able to guide chondrogenesis and matrix deposition. And this is probably due to the increase in the hydrophilicity of the materials and also to the presence of carboxyl groups and aldehyde groups that lead to this basically covalent attachment between the growth factor and the PCL mesh. Another direction that we are basically, and this is really my last slide, that we are trying to look at is to how can we further advance the nozzle-based techniques in order to create microenvironments around the cells that basically represent that microenvironment. Typically, we look at extrusion-based techniques, which are techniques that have a limited resolution in terms of the filament size. So typically, we can achieve 100 to 200 microns of filament size, which basically brings in quite some limitations on controlling the architecture and then environment, or the niche of the cells in our native living tissues. So we have been looking at techniques like electrowriting and then advancing it towards the incorporation of living cells, because we think that this increasing the resolution, and of course not for all applications, but for some applications, will provide us further control over the microenvironments of the cells, which has some limitation or some advantages in terms of guided tissue regeneration. We are also looking at this technique as a tool to basically further study what are the mechanical forces that induce cells to lay down collagen. And we believe that the shear forces and the tensile forces that we expose cells in this, what we call cell electrowriting process, are basically interesting and the foundation for laying down aligned collagen. So basically what we did was look at basically two hydrogel bioinks, in this case based on two polymers with different gelation chemistries, so gelatin and silk fibrin. And for the gelatin, we look at the Norbergen modified gelatin that can basically form an hydrogel through step growth polymerization in the presence of, in this case, multifunctional thiolene crosslinker, and unmodified silk fibrin that basically undergoes quick photochemical crosslinking due to the presence of tyrosines in its protein structure. And basically, we also, to accelerate the cross-linking reaction, because it's paramount if you want to print with living cells, is that you have a quick cross-linking reaction. So the cell, the hydrogel filament cross-links and solidifies immediately, and it's not drying out, because that will basically lead to a low cell viability. We combine it and we explore the use of two components for the initiator system, in this case, ruthenium and sodium persulfate. And what was very interesting to see was that, and then walking you through, is that the two different hydrogel combinations allowed us to achieve the rheological properties, in this case the viscosity and the speed of cross-linking that we needed for creating a stable jet. And the addition of PEO was also crucial here to achieve the viscosity that was essential. At the central column of this slide, you see that we could, as I mentioned, achieve a stable jet formation, but that was after quite some tedious work on exploring key instrument parameters, or the influence of key instrument parameters on jet formation. In this case, we look at the voltage, distressing pressure, and collecting speed in order to get straight fibers and being able to actually control the architecture of the printed constructs. And then by doing so, we could achieve control laid down on fibers with different fiber spacings. We went down to 400 microns with printing accuracy of around 80%. So this printing accuracy means the deviation between what you designed and what you printed. And below 400 microns, there was a significant decrease of printing accuracy. So we started to lose the ability to lay down patterns, or to lay down fibers in a controlled way. And this was more evident for the silk material. The next step was to basically incorporate the cells and then look at cell viability. And what you see here is basically a comparison between extrusion-based printing with cells encapsulated and our cell electrowriting process. So in this cell electrowriting process, you create filaments with isolated cells, forming a nickel-like type of filament, somehow aligned, but this alignment still have to be further evaluated, while in the extruded printed filaments you see an heterogeneous distribution of cells with no preferential organization, of course, with a higher cell density. And it was also interesting to see that we maintained the cell viability high. This was higher for the gelatin-based inks, and it was somehow some changes on the cell circularity within the cell electrowriting, which basically induces or tells us that there was somehow these shearing forces stretching the cells without basically altering at the cell level some aspects that we don't want to alter, but this of course requires further understanding. But what you see is that you can print with living cells, cell viability is high, and then somehow we are, by shearing the cells, aligning the cells. So as a few concluding remarks and highlights that I would like you to take home from my talk, basically I show you that patterning microfibers from natural derived polymers and synthetic, of course, it's feasible, but certain questions remain answered, so including how shear, tension, and confining forces on non-fibril structure formation are influencing, and this I'm referring to the extrusion-based printing process. I also show you the electrowriting of cell laden microscale biomaterial fibers. It's feasible. I show you, and sorry that I'm not let me just quickly look here. So starting again from the beginning, basically I showed that some fiber-based scaffolds holds significant potential for regenerative medicine, and that extrusion printing in sacrificial support baths enable the creation of cross-linked fiber collagen structures, and that shows some collagen hallmarks, in this case the bending pattern and fibril organization. I also show you that the electrowriting offers an improvement on resolution when compared with traditional extrusion-based methods, and it's applicable for to both synthetic and natural polymers. I show you that the electrowritten fiber-based scaffolds have the potential to imitate, and here it's really imitate, not resemble, some complex tissue and organ functions. And I show you examples to restore the contractility of cardiac muscle and filtration removal waste products in kidney. And then, as I started from this talk highlights before, so apologies for that. And the last highlights that I wanted to take out is that patterning of microfibers from electropolymers, as I mentioned, it's feasible, but a couple of questions remain unanswered. I also show you that we can advance electrowriting to aqueous-based laden bioinks printing, and that the convergence of electrowriting with some techniques like cold plasma treatment enables covalent attachment of proteins, which offers great opportunities to guide endogenous tissue regeneration. And with this, I would like to conclude my presentation. Thank you very much for your attention, and then for staying on that side. And then I'm happy to answer some questions that you might have.
Thanks so much, Miguel. Really fantastic presentation. Really appreciate you taking the time to talk us through your latest research. Really some impressive work there. So just a quick reminder before we get started with the questions and Q&A session, please feel free to ask questions on the question section on the GoToWebinar interface. So we've got a few questions around, I guess, PCL and melt electrowriting. Maybe before we go ahead with those, Miguel, I think maybe leave your presentation on for now, just in case there might be some questions that relate to your data.
Right.
I'll go through them one by one as well.
Can you see it still? Or?
Yes, we can see the beginning now. All good.
Yeah.
Cool. So first question. Hi, and thanks for the interesting presentation. I want to use MEW for PCL. How does the molecular weight of the PCL matter? And if it matters, what molecular weight would you recommend to use?
So I don't know by heart which molecular weight we are using. It definitely matters because viscosity of your polymer material that you are writing, it's quite crucial. Typically, we work with medical grade polycaprolactone from Corbion with a specific molecular weight. You can take this from our recent publications and later publications. I think we all use a very similar molecular weight. So it definitely matters because the rheological properties of your material are quite important, and typically we want to print with highly viscous materials that prevent dripping.
And is there any published protocols for how electrowriting is performed, and particularly with PCL? For someone who's a novice, might have a PCL or a metal electrowriting machine, but hasn't really started with PCL writing.
Yeah, I can. If you drop me a line, I can guide you or indicate you some references that basically I don't know until which detail or which extent they described all the aspects there, but definitely it can provide some starting points, and then I'm happy to share the information that we have on the formulations that we use and the PCL material that we use.
I believe, if I'm not mistaken, that Professor Paul Dalton from, I think, Oregon at the moment, he also published a number of papers, of course, and also potentially protocols around actually the development of your own open source melt electrowriting machine, which I think is a very cool approach, and also some protocols for how to get started with PCL printing. Another question also relating to the MEW, and I think there's some information, of course, in the literature available, but maybe you can quickly comment on that as well. How can we optimize fiber diameter and pore size in MEW? So I think you showed one or two examples of different inks and materials that you were printing. It seems to be a fairly, I guess, trial and error type of technique that you have to do. You have to change voltage and velocity and things like that, and then observe the jet. Maybe you can just comment on that a little bit.
Yeah, so I think for the synthetic polymers there is more literature already showing, and also we have shown, but work from Paul and also colleagues in Germany have shown how, for example, pressure influences on fiber diameter, and then they show that how they can control on fly the and then on fly, I mean during a printing process, by just adjusting the dispensing pressure, they could adjust the fiber diameter. Of course, other parameters in the process influence on the fiber diameter, like the voltage and the collecting speed, but the one that influence the most are really the dispensing pressure. For natural polymers, it's more indeed trial and error approach. We are now following a more systematic way of a design of experiments to identify which parameters influence and would allow us to control fiber diameter, but it's still mostly based on a trial and error so far when we refer to natural polymers. In terms of the pore size, of course, we specify by design what is the interfiber distance that we want to achieve. There are some issues that below some interfiber distances we cannot put fibers together, and that has to do with the remaining charges that are trapped in the fibers. Here the literature is a bit contradictory. Some defend that indeed remaining charges are the reason for a repulsive effect, and the fact that we cannot put fibers below 50 microns, let's say, together. But others do not attribute to this issue. So there is some work needed here to see how we can bring low, and here I'm saying from the top of my mind, the 15 microns distance between of pore size. It's the one of the limits, but here it requires more work to understand how can we go low. But I would say if you want to control the pore size, until 100 microns is basically just designing your construct that dictates the pore size.
Yeah, that's correct. Also from our own work, we've noticed that if you print very thick scaffolds, let's say with a certain pore size of around, let's say 200 micrometers, the bottom maybe 50, 100 layers seem to be very precisely deposited at 200 microns pore spacing, so the target spacing, but it does become a little bit less organized the higher you print.
Yeah.
Our hands. Another question here is, hi Miguel, thanks for your presentation. A question is, how different is the shear thinning in electrowriting compared to typical extrusion-based bioprinting?
Well, keep in mind that in the electrowriting we are basically combining two forces. So the electrical force with either the pneumatic force or the mechanical force that you might use to dispense your material. These will lead to shear forces inside of your nozzle. One has a contribution of two forces on extrusion-based printing, just basically due to the pneumatic pressure or the mechanical system that you are dispensing your material. I think the shear thinning, you are referring to the material properties that you achieve, right? And how is that different between one and the other?
Well, the presentation is coming from one of the adaptees. Maybe we can just add that information as well. But I think from my understanding is, obviously in extrusion-based bioprinting, you need to optimize the bioink to display a very strong shear thinning behavior. So you have something that is maybe a gel initially, and as you apply force, the molecules align and it becomes liquid. And as soon as it leaves the nozzle, it becomes solidifies again because the polymer chains, I guess, are tangling up again. And my interpretation of the question is whether this is also an important property for the material to be extruded or printed using MEW.
Yeah. No, I agree. Yeah, if that is the question, yes, it's also an important property. Of course, for MEW, you need to consider other properties, as I mentioned, like the conductivity, that are also important. And then here, the shear thinning, and I would say the speed by which your fiber solidified, it's way more important than in extrusion-based printing, because since you are combining two forces, you are accelerating your jet 10 times faster. So you actually need your material to solidify way faster than in extrusion-based printings. And then, of course, in extrusion-based printing, there are all tricks of printing in a support bath that basically allow you to print also with liquid-like formulations that does not have any shear thinning behavior.
Fantastic. I had a couple of questions as well, Miguel. So early in your talk, you showed the addition of polyethylene glycol to, I guess, a collagen-based hydrogel and how that influenced collagen fibrillogenesis. And if I understood correctly, you found that addition of PEG essentially increased the speed of fibrillogenesis. Is that correct?
Yeah, indeed. Yeah.
Can you maybe just explain how the mechanism works? I think you did mention it's a crowding agent, but I wasn't familiar with that term or how that functions. Do you mind just going through it one more time?
Yeah, so basically what we know is that the fibrillogenesis takes place in the ECM environment in the presence of other macromolecules, and some of them participate in this crowding effect. And it's basically just by occupying space and then further confining the polymer molecules. And then occupying space and then excluding water leads to a further confinement of the, in this case, collagen molecules, and that speed ups the process by which you move from collagen molecules up to fibrils and then fiber bundles. So it's really just occupying space and confining the material, which what we believe that leads to this fast fibrillogenesis or faster fibrillogenesis time.
Basically, if you have a PEG-based solution you're adding collagen, your expectation would be that the collagen is not homogeneously distributed throughout, but there would be basically, on the nanometer scale, pockets of collagen where they can.
Okay. Exactly. Yeah.
I think that makes a lot of sense. You also mentioned about the cross-linking of collagen. So there's a couple of enzymes that are typically involved, transglutaminases, lysyl oxidases. Is that something that you can potentially just add on later to your scaffold to enhance the mechanical properties?
You can. So what was very interesting for us to see, and I think in this slide I only mentioned very briefly, is that these printed constructs were stable up to 2 months. And then we basically believe that there was some sort of native-like cross-linking going on in there that did not require any addition of cross-linking agents like the transglutaminase that you were mentioning. So here was really just playing with the pH and the ionic concentration of the bath, and then of course again the confinement that led us to a stable collagen network formation. But of course, I would be very hesitant on saying that they are fully cross-linked and how. But this is something that we are now investigating.
Oh, fantastic. Another question I had as well. So in effect, a little bit later on the slide, you showed that the alignment of collagen fibers obviously is very important in different tissues. In the hard tissue in particular, I think that's very a complex structure, and it's so important for the function of the tissue, really. I'm curious, maybe this is a very in-depth question, and as a tissue engineer, you might not know the exact answer, but do you know how that, from an embryogenic point of view, how that even functions? How developing embryos and tissues can really control the deposition of collagen fibers in a certain orientation? I'm just curious. I'm not actually sure how that functions.
Well, that's actually a very interesting point, and we have a project that we somehow look into that embryonic heart development. And what we know is that basically it starts with a tube that then folds and then starts making the ventricles. It's a bit of a chicken and egg question because we don't know which dictates what. First, is the matrix that influences the alignment, or are the cells that somehow when laid down they get aligned? So we are now looking at different stages of embryonic development, looking at the stiffness of the ECM that we get from chicken embryos, study that, and then look at the influence on seeded progenitor cardiomyocytes. But this is a very important question, and I think it's a bit of the chicken and egg question that I honestly don't know. But it could be quite important for the design of materials.
Absolutely. I think that's a common theme, that in tissue engineering, we try to often aim to replicate the final tissue as it is in an adult without necessarily going through the same developmental stages that we would find in a developing body or embryo as well. And maybe key to success is really to replicate that whole process a bit better as well in future.
I think that is, if I can just add one note there, Christopher, I think that it's a very important point, but for the sake, when you have already a matured heart that you basically want to restore contractility, I think there you are already a step forward, right? Because you want to, that you want to restore contractile force, so you need to get that organization. So there is not much time, I believe, to go through all the development steps. But I don't know.
I agree. I mean, it can take many years, right, to develop full. Absolutely true. And particularly for cardiac patches, I mean, you would expect that in that scenario, you might have a certain area of the heart that's damaged that requires repair, and you deliver a cardiac patch with a well-aligned, collagen fibrils, and also the cells are well aligned. If you place it in the right direction, it would make sense to be able to just restore that without being able to fully, recapitulate the heart and the alignment of fibers there as well. Cool. I've had one or two more questions, if you don't mind. So towards the end of your talk, you were talking about the atmospheric plasma, which I thought was really interesting, and how you can actually organize and retain growth factors. Really cool technique. I've actually not seen that before. Really cool. I was curious if you can actually spatially control growth factors using that approach. Like different tissues, and particularly when you think about organs, maybe there might be the spatial location of different growth factors might be really important to really generate, let's say, functionality of an organ rather than just a simple a single tissue. Have you looked at that at all?
Well, it was one of our goals at the beginning, but somehow the project was already going quite too long, and there were so many challenges to tackle that we dropped that part. So I would say it's possible. It's something that we started investigating, but we didn't continue. What we showed was that in the out-of-plane direction, we could somehow create a gradient, not very controlled, of TGF-beta distribution. So the one that was exposed to plasma at the surface had a higher concentration, and at the bottom a lower concentration. But these were really just preliminary tests that we couldn't control or investigate further, but it's definitely a way to go. I think in plane you can definitely have regions or functionalized or non-functionalized or activated with 30 minutes, another just 10 minutes, and then create gradients that way. But it's something that we didn't explore, but it's fairly interesting.
Cool. Last question from my side as well. In the last application you showed today, which I think was also really, really fantastic, so you used the gelatin-derived polymer, I think gelatin nobony functionalized, and you've melted electrowritten this, particularly also with cells. I mean, really nice work. I was curious, which cells did you use for that application, and whether you saw any elongation of the cells over time. So I think you had a really nice picture where you could see the very fine fiber and then a cell basically embedded in that fiber, which I thought was really cool. Did you see the cells elongating over time and stretching out?
No, that was actually a limitation that we had. So we did these stainings over 3 and 7 days, and what we observed is that it's still a challenge that we have to solve, is that the fibers dry out over time, and then since they are so small, they disrupt. So this keeping these constructs over longer culture periods, it's something that we need to address, either by increasing the polymer content or then enhancing the cross-linking of the material. We used MSCs here, and then the idea was just to use a cell that could then be differentiated on different tissue-specific cells. But long-term cultures is something that we need to address. And we seem to have some kind of elongated cell, but these are again preliminary tests that will require further work.
No, really nice. And is this work already published?
Yes, this one it is. Yeah.
I could read that paper as well. Really nice work. Well, thanks so much, Miguel. Really fantastic presentation. I think we'll wrap it up here and from the Q&A, unless there's any last-minute questions from the attendees as well. So yeah, again, thanks so much for your time. I'll just turn off your presenter view now. Really appreciate you joining us today. Also to our attendees, thanks so much for joining us today. So before we wrap up, just, if you're not familiar with Gelomics, make sure to check out our website as well. We've got lots of content there, lots of materials, really cool products as well. If you're keen to test anything, let us know. We provide free product trials as well, so be more than welcome to test and trial our products in your own laboratories, in your own spaces as well. So this is, we are having a webinar series as well. So this is the 3rd out of 4 webinars this year. So there will be one more webinar announced towards the end of the year, most likely in November or December this year. So please keep an eye out, everyone, for the last webinar. And also we'll be making available a recording of today's webinar as well on YouTube, and also I think on an on-demand platform as well. So yeah, with that, again, thanks so much, Miguel, for taking the time to present to us today. Really fantastic work. Really excited to your future impact as well in the field. So really congratulations on everything. Great achievements. And yeah, thank you for everyone as well for attending today. We really appreciate your time and your engagement, and really look forward to connecting with everyone again in the next webinar. So thanks everyone, and talk to you soon.
You're welcome.
Thank you.
Awesome. Thank you very much.